All right, I think we'll get started. So it's my pleasure to introduce Professor Michelle Digman, who is our department chair of biomedical engineering. She's also the PI of the Laboratory for Fluorescence Dynamics of the LFD, which you were able to see last week. And she's going to talk a little about her career path. So I won't talk much. I won't say anything more about that. But one thing I do want to just say and just to thank Michelle for is one of the things I find most remarkable about her is that she is tirelessly selfless. She's held so many service roles here at UCI. It would take forever to take the whole time basically to go through them all. But also, whether it's mentorship, whether it's just, basically anything that she's asked to do, she jumps right into it. She doesn't hesitate. And so with that, she actually has her own short course this week. And it's 29 participants, so double the size of ours. And it's this wonderful program. It's I think 14 years now and counting.
Yeah.
It's
Yeah.
basically with high school students and community college students to give them a research experience over the summer in biomedical engineering. And there's some really nice stories that have come out of that program in terms of where they've gone from this. And so, with that, she also does she's an expert sword wielder. So she keeps us in line in the department that way. She gets us to do things. So with that, let's welcome Michelle.
Thank you. Thank you, Bernard. Thank you so much for having me. And I am really excited to talk to you about some of the research that we're doing in my lab. So let me see here about you. How many of you are from the East Coast? Okay, excellent. Midwest. All right, I'm from the Midwest and West Coast. All right, and from outside the United States. Any internationals? Okay, wonderful. Well, welcome to UCI. I know you've been here for at least 6 days already. This is the building where my lab is, also the same building where the biomedical engineering department is on the 3rd floor, and you've been there last week, right? The LFD. And the LFD has, sorry for the wording there, customized microscopes. We have ones that we've actually designed ourselves. So those are the ones that are very generically named M1, M3, very, creative names. M is for microscope. Microscope 1, 2, 1, 3, and 5. And then of course we have the diver, which you see the image of the diver here, or this one's M5. I don't think I have the image of the diver, but it looks very similar. It's all encased in this kind of black shield. We also have commercial microscopes. We have the ISS Alba STED system, the LSM 880, the 710, the Olympus Fovi 1000. We also have full field imaging systems. And then in my own lab, I have a few other microscopes that we use for imaging. So as you can see, everything that we do is fluorescence imaging, fluorescence type imaging. We have, besides the core instrumentation, we also do individual training. We have, this is at the heyday when the LFD was, let's say, the biggest that we've ever had it. We have 13 PhD students between Professor Enrico Gratone and I. He recently retired a few years back. And then we have visiting students, scientists, and engineers, and I still get visitors from all over the world. As a matter of fact, I had a visitor in May from Argentina that came. And I'm going to be getting a few visitors from Italy this year. And we do a lot of outreach workshops. We do our own LFD workshop. This year, we're actually combining forces with the AIM workshop at UC Berkeley. They do their workshop in January, and we do ours in October. And we said, hey, why don't we join forces and just do one big workshop, and we'll alternate between Berkeley and UCI. So we're doing the first combined workshop at UCI in January, which is the week before SPI meeting, the Photonics West meeting. And that's generally when Holly Aaron had hosted her workshop. And then next year will be at Berkeley. We also worked with other centers on campus to work with them to train students. We do our USIBR, which is this week, is our outreach program for students. And as Bernard mentioned, we have I had over 90 applications to our programs, only a week, and we really do a condensed session. But I think the students really enjoy it and love it. We work with Cosmos and some other programs across campus to do our dissemination and training. So what are the imaging technologies that we mostly work with at the LFC? You've heard of the phasers, you've heard of fluorescence lifetime imaging. We also do multiphoton excitation, second harmonic imaging, third harmonic imaging. We do fluorescence correlation spectroscopy techniques. And this is kind of one of those derivatives of FCS. And what you're watching here in this movie is we're inducing double strand break with a 2-photon excitation laser. Protein that is tagged to the green fluorescent protein gets recruited to the site. And then using pairwise correlation function analysis, we're able to build tensors of the molecular flow. So essentially, the way that the molecules diffuse into a given space in a given time. So time and space correlations, it's like similarly to you sitting here in your seats. When it's time to leave, all of you are going to exit out the door, right? So using pairwise correlation functions, we can actually analyze which route you are most likely to take in the exit. That's what we're mapping here. And to do this, you need fast imaging systems. So we have TURF system. We also have very fast raster image correlation spectroscopy systems. And we can determine the diffusion rate. This is the autocorrelation function that gives us the direct diffusion time of the molecules in a given space, so within the volume of excitation. And then from the same images, we can map the protein dynamics. So we can map monomers, dimers, tetramers, higher order aggregates based on the variance of the fluctuations. So the phasers is something that we contributed to the field of FLIM. You've probably heard Alex's talk on phasers. He was a former postdoc in the LFD. And I just met him on the way out, so that's what I know. So FLIM is one of these amazing techniques that you could use to identify molecular species, right? You can do fluorescent species, you can do autofluorescent species, and the phasers gives us a way to actually very, very nicely without having a huge knowledge, right? You can just do it immediately, see the results, and analyze the data by graphical representation of the lifetime data. And so, let's see here, you can use the phasers to quantify the FRET efficiency. This is an example of a protein called RAC. RAC is a protein that is a GTP bound, it undergoes GTP to GDP binding, depending on the type of state of GTP or GDP. And the sensor that was made by Klaus Hahn and many others uses a donor and acceptor pair. And when the cell undergoes, let's say, a positive RAC activity, we see a positive RAC signal. And most of the RAC protein is activated at the lamella edge of the cell. That's because the cell is going to be moving and GTP proteins are needed for that motion. So we also developed the phasers for something called the Z phasers. This is essentially a poor man's super resolution way of imaging. You don't happen to have super resolution systems in your lab, but you have confocal microscopes. What you could do is you could image, let's say, the stacking of proteins. And this has been very useful for looking at focal adhesion, let's say stacking. And basically what you see here is the Z stack of 2 different types of focal adhesions. And we did this in real time, so you can actually get a real-time movie of the Z stacks. And you can get very fine resolution between the distance of, let's say, talin and integrin, or integrin and paxillin, or whatever protein of interest that you're interested in longer in this position. And of course, hyperspectral imaging has also been very, very useful. The first one that transformed the spectra into phasers was Hans Gerritsen. And we've done it as well. So we transformed the spectral emission into phasor space, and that allows us to quantify separate the colors. I'm going to show you some examples of that. So the phasors is not new. The phasors have been used since the late 1900s, well, 19th century, specifically in electrical engineering. So if you have an electrical engineering background, this term is very familiar to you. And yeah, so you have, you can take any sinusoidal signal. This is an AC, for example, signal, and you can transform it into phasor space. So basically what you're transforming is you get, let's say, 2 points on a plot. You get the vector length, right? That is defined by the modulation, and then the angle is defined by, in whatever case, a phase shift, right? So the phase and the length of the vector is the modulation. And so you can transform this into phasors. And last week I was in a really cool meeting because these are all people that come from chemistry background, physics background. They're asking, why didn't you use, a different transformation? Why did you use the phasors? And the phasors were traditionally used because we were in Enrico's lab many years ago. They were using the frequency domain for FLIM signals, and they were doing it with solutions. They were not doing it in images, and you can get very good quantification of the frequency space, the frequency domain data from FLIM into the phasors, and that's just traditional, right? Some people say, why don't you explore transform and look at frequencies? Sure, you could do that, but how do you isolate linear combinations of 2 or 3 or 4 unless they're separated somehow with frequency, right? And sometimes it's not as easy as it seems, but if you want to try it, I highly encourage you to see how well you can separate those signals. I haven't tried it myself, so I don't know how it would work. So Andrew Clayton, Quentin Hanley used the phasors. They call them the AB plots that they published in 2005. Bob Clegg and UIUC. This is where I did my postdoc, and his student Glenn Redford, which I overlapped, so I knew Glenn, used the phasors. They call them the polar plots. And then Enrico and I, there was a group from Italy that came to the LFD at the time, and they were really wanting to measure the FRET efficiency. So we decided to do FLIM images. This is the first time that images were converted into phasor plots. All of this other data was from solutions. So they were just converting the data to solutions. So we know that fluorescence is a really important signal because it has high specificity. You could label specific regions of the cell. You could get a tumor tissue and label the cells. You can identify each cell individually. You get a lot of information from an image like this. And of course, you can use many, let's say, variants of the fluorescent proteins. You can also use combinations of dyes. You can also engineer these genetically encoded proteins to undergo sensing. So for example, you have a donor and acceptor. They have their FRET pairs because they have less than 10 nanometer distance between them. And when a molecule binds to that binding site, they are resonance energy transfer occurs, and you can map that in a really image. So fluorescence is very important. However, it could be limited to short time length scales. And what I mean by that is that you can't continuously image this with laser beams, right? Because eventually you're going to get phototoxicity, you're going to get quenching, you're going to get photo bleaching, you're going to get a lot of different photophysical effects. So in my lab, we have been working with a group on campus, Jennifer Prescher's lab. She's a chemist. They've been synthesizing different types of substrates for bioluminescence. So I'm going to transition you from fluorescence imaging for the minute to bioluminescence. Now, bioluminescence is laserless based, right? You don't require any lasers at all. So, for example, you have, what is bioluminescence? You have basically it's an enzyme, enzymatic reaction that occurs in the firefly. If you've ever seen one, those of you from the Midwest, you might have seen it. West Coast, not too much unless you go up to the parts of like Oregon and Seattle, Washington states maybe. Fireflies have this naturally occurring emission of light, and that's because they express the protein or the enzyme luciferase. Luciferin is the substrate that binds to luciferase in the presence of ATP, oxygen, and magnesium. And when it becomes oxidized, this substrate turns into oxyluciferin and it emits light. So that's the beauty about bioluminescence, is that this reaction will happen in naturally emitting light. So you can actually image these really nice emitters for very long periods of time. And they'll be luminescent, so for long periods of time. So for example, you can take a mouse on day 12 and look at it day 17, 21, 22, 31, and all you have to do is add the substrate and it will illuminate. So that's the beauty of bioluminescence, is that they're pretty good. The only properties with the firefly luciferase is that it is actually pretty weak photon output. It's described as having 40 times less brightness than the new reporters that I'm going to show you. So the engineer new bioluminescence reporters are called the brighter reporters or the nano lantern reporters. And essentially what these have, they're also genetically coded. So you have the protein DNA of luciferin or Luke that has a binding site on its surface somewhere, or its structure for its substrate. And now when that reaction occurs, it will emit light, but when the emission of the Luke reporter emits, it will transfer its energy to an FP. And you can swap out the FPs depending on the conformation of Luke and the substrate that you use. And you can make them either blue, green, and yellow. So the FP nanolantern would be the GSP, YFP, CFP, whatever you want on there. So you can multiplex many reporters at the same time. So now you have an advantage of swapping out the FPs, enabling you to have much broader range in detection. They're much brighter. So that's the nice thing about bioluminescence. They're very, very bright. They're like about 40 times brighter than those, than the original firefly ones. And they have a very broad spectrum. So we started working on this project with Jennifer's group when actually Professor Vasan Van he got an NSF grant for a training grant. So this was the IGERT grant, and one of the recipients of this grant was this graduate student, Miguel from Jennifer's group, and I was teaching a class specifically for the group of students who have gotten this training grant. And I was going through the FLIM lectures and spectral lectures, and Z came up to me and said, "We do this transformation of the phasers from bioluminescent signal." And I said, "Sure, I think we can. I think it would definitely work. We just have to have a way to detect it." And so immediately, like a week later, Z shows up to my knocks on my door, my office. He's like, "I'm here, I'm ready to take the images." And I'm like, "Oh my gosh, we need to actually have a system to actually detect the bioluminescence," because all of our imaging systems are laser-based, spectroscopy-based. So indeed, we had to rearrange our imaging system. And with another graduate student and a postdoc in our lab, we decided that it would be very simple to engineer this part of the system so that we can detect bioluminescence and thereby transfer the spectra, and this is what you're seeing here, into the phaser space. So how does this work, right? So you have, let's say, let's wait till this thing starts over again. It's going to do it in a second. So imagine that you have a very sharp emission peak, right? The width is only 10 nanometers. So for whatever wavelength that is, the phasers, then the length of the vector is the width of the signal, how broad that is. That's called the modulation for us. So we transform the length into the modulation. That will be, see how it's broader, it gets closer and closer to the center. Now, the wavelength, so between say in this simulation, it's like 400 to 800 nanometers. You start at a given position, and then the phase will be determined on the wavelength, the center of the wavelength. Okay, so then this S and G components is all you need to plot the pixels from your image onto the phasors. Okay, so that's how we get the phasors from this. And the beauty of the phasors is that you can actually calculate the linear combination of, let's say, 2 dyes, 3 dyes, whatever you have. In this case, we have bioluminescent reporters. So if we have 2, linear combination of A and B, or B and C, or C and A. If you have 3, you can calculate the linear combination of all 3 inside that triangle. And then you can calculate the fraction as it's doing up there at the top, the fractional contribution of A, B, and C. Okay, so going back to the luminescence technique, so we have a problem. The problem is that with a lot of these bioluminescence reporters, they overlap a lot, right? If you look at the spectra right there, they're overlapping. But when you transform the data into phasor space, they completely separate out. So that's the nice thing, is that it's not a linear unmixing technique. So if you go to the microscope and you have a spectral detector, and then you try to extract the colors from each of your pixels from this overlap spectra, it's going to ask you to load a file of the known spectra of each dye that you have. So it will linearly, let's say, unmix those colors, and then you get an output. So here, there's no linear unmixing. This is just a nonlinear process because it's just the spatial position of the phasors. So as I mentioned, we had to reconfigure our microscope. So what we did is we used a turf microscope, but not in the turf way. Obviously, we're not going to use lasers. But the emission signal then, once the chemical reaction happens, the biochemistry that happens, there's a split here. We have a splitter that goes to 2 cameras. Now we are losing a lot of light using 2 cameras, but how do we make this spectral, right? We don't have money to buy these fancy looking spectra cameras. So instead we use sine and cosine filters. Now, the sine and cosine filters are something that my lab didn't invent. It was something that Enrico started in his lab with, I don't know if you met Sasha. So they used actually the first time they used the sine and cosine filter, they bought it from Amazon for $5. And people are laughing, saying, you're never going to get spectra, right? So in fact, the spectra was not perfect. It was actually all. So they had to correct for sine and cosine, right? So you should get a perfect, this is not a perfect sine and cosine. This was hand drawn, so it's a little shaky. But if it was perfect, you should be able to correct for each one of these points along the sine and cosine graph. And then you can represent this and transform this into the phasor space. So now we got a little bit fancier. Yeah.
So so you're doing this in wavelength space. So then the sine and cosine, so it's one cycle. That's what Alex had told us about.
Yeah.
And so that then means that you're kind of fixed in terms of what's the shortest wavelength and what's the longest wavelength based on like that becomes fixed then.
Yeah,
Right.
and if you have perfect sine and cosine filters, so we have custom design ones that go exactly from 400 nanometers to 700 nanometers. And these are the feature of one period gives us the first harmonic, and that's all we need to that. So we can obtain the entire spectrum just with the sine and cosine filters. But we do need a reference arm, a reference channel we have to split the viewer into 2 for each camera. So now we're splitting it into 4. So we lose a lot of light. And in fact, for proof of principle, we just wanted to see if we saw something with the brightest bioluminescence reporters, and we could. So we used the nano lanterns. They were the brightest ones that you see. And so seeing the combination of 3 cells here, 1 cell, excuse me, is expressing the YNL, the other cell is expressing the OENL, and they're spatially separated in the phaser spot, and this gives us hope that this is going to work. That was our very first image that we got with the reporters. So, any other questions? Yeah.
Yeah, very dumb question because I'm not quite familiar with this. But yeah, I see, as Bernard said, that this is like in the spectral space, let's say. But we saw in the lab the phaser approach for time domain measurements, right?
Yeah, yeah.
So I wonder if you can use this bioluminescent phaser approach with the time domain measurements.
Time domain. Right. So the time domain was used to have a reference with a clock, right? So you
yeah.
have the repetition rate of the laser is your clock, and then you're collecting photons as a function of windows. You have 12.5 nanoseconds to collect the photons, right? Within one, let's say, pulse, and then the next pulse, and then the next pulse, right? So one pulse, you collect the number of photons, another pulse, collect number of photons. So that's the time domain.
Yeah.
So in this case, we're just looking at emission wavelength. We're not
Yeah, you
looking
don't
at
have
any
any idea of
time
referencing.
pulsing or anything
Mhm.
like that. It would be wonderful to have it timed,
Yeah.
because then we could do fluorescent, like
Mhm.
emission lifetime. Unfortunately, we can't synchronize anything with the reactions.
Yeah.
Good question.
And maybe a second question, do you have to take into account any instrument response function or something like that?
Right, so that's why we need the reference
Okay.
window for that, so that we could identify kind of an instrument response function for our reference. We need to be able to calibrate so we know exactly where to start our phase for the emission signal. Yeah, so we get a little bit of direct light signal.
And just to be clear, those to the things to the right of those filters, those are spectrometers, correct?
So this is this cosine into one part of the camera, and then the reference, there's no filter, right? So is that what you mean? There's no spectrometer anywhere,
Oh,
right? Yeah, you could put a spectrometer so that you could just calibrate your function, but yeah,
okay.
we don't have any spectrometer. There's no spectrometer here, right? These cameras are not built to actually like do spectra, because they've never expected to have a spectra, but they collect within the range of the visible spectra. Yeah.
Yeah, that's my questions. But you do need a spectrometer to measure the sensitivity, like the transmission spectrum, and correct them, right?
Yeah, so that's why we have the reference window, right? I mean, we don't have any spectrometer here.
But
It's so simple.
yeah, because you just said like the transmission spectrum is kind of noisy, right? And you had to calibrate that.
Yeah, yeah. So we have a reference window with no filter at all. That's our reference window. Yeah. That's our instrument response.
So reference window to calibrate that noisy spectrum.
So it'd be, we would collect all the light that is being sent, right? Because here we're splitting the light, so using the sine and cosine into like one segment of the spectra to another segment of the spectra, right? And this reference window collects everything. So it's like, this is our instrument response function. Think about it that way, to
Okay,
calibrate the phaser.
Can I understand using this imperfect cosine and sine like filters, the phasor space is not the perfect phasor space. Like that, do you really smooth those curves to be
Yeah,
perfect?
these curves are very smooth. They're just handwritten right there. You can probably redo those. Yeah, they're perfectly smooth. They are, so the feature period of each function is very perfect. So we use the first harmonic. So essentially, if you put these sine and cosine together, it makes a perfect circle.
Yeah. So yeah, so can I say although I don't know what's the algorithm, but by having this reference beam, you can calibrate this imperfect curves into that
Yes.
perfect curves.
Yeah.
So that the phasor space is the original phasor space you
Yes,
intended.
yes, that's correct. Mhm.
Okay.
Yeah. Any questions?
So the sine and cosine filters are like made so that within a range of your wavelength, it transmits kind of like a sine
Mhm.
and then sine wave, basically. If you have like if your min wavelength is 400 to 700, it creates that sine curve or cosine curve and then lets those wavelengths pass through.
So it will collect, yeah, it will collect the signal, so the light, starting at 400 nanometers, right? Whatever that is, and you have whatever intensity and however broad that spectra is, right? In the sine, which is like the 400 to, I'm not sure, like 500 something. And then cosine will start at 500 to 700, right? So
Or like absorptive filters.
Hmm?
Kind of like absorptive filters. It like absorbs the wavelength.
Ah.
Or my
yeah.
first time hearing.
Yeah, I guess, in this case, we really wanted perfect sine and cosine filter to make a perfect circle. Because that's you get the immediate data, all these pixels are transformed. So that, all of these points are the pixels that are in this image. Right, so you have an S and T coordinate for each pixel. Yeah. Any other questions? Yeah.
So, yeah. So for the filters, like cosine and sine filters, how important is it to have it? Because it's a filter through a spectrum. How important is it for the resolution of each of the points of that sine wave to be? Does it need to be
It needs
1
to be a perfect
nanometer?
sine and cosine filter. Yeah. I guess it's the question, like, how good is the light that goes through the filter that,
Oh, I was thinking more of so is it more like it has to be some sort of discrete numbers. For an example, if you have like 400 nanometer, you attenuate that much. 401, you attenuate that much. Does that resolution matter?
oh, I see what you mean. Yes and no. It depends on how bright your signal is. If you have very intense signal, you're gonna have a very good, let's say, if you get very noisy signal, your pixels are not clustered, right? So for the image, right? I mean, it depends on the question. If you want to know, the level of resolution within your image, right? And you're saying, okay, can I trust that that one spot is actually yellow and not red, right? That one pixel. So for that level of resolution, we haven't looked into that great detail. We're suspecting it's pretty good in terms of that resolution. I mean, you could see some graininess here that could be because some of these points are not clustered in that like main spectra. They might have a little bit broader spectra, or they might be due to some environmental changes, the probes changing. We're testing that now to see if these probes actually change spectra in those environments. We don't see too much of a change, but if you have more noise associated, it'll look broader. It will look broader. Yeah. Mhm. Okay, so now we characterize the cell in culture with different bioluminescent reporters. We have blue reporters, cerulean reporters, green reporters, yellow reporters, orange, red, and Lumi scarlet. So we are able to identify the spectral position for each one of these spectra. So if you look at here, right, the spectra is really broad. So if you combine all of these, it's going to look one big giant mess, right? Right now, they're spatially separated. They're in different cells in different culture dishes. But now, if you mix, let's say if you take the cells and now shuffle them and culture them, you can identify what cell is expressing what reporter, and they are spectrally separated, right? So I can see that Lumi scarlet is here, RENL are the red ones, yellow are YNL, and so forth. So you can identify which cell is expressing what reporter. But that's not all. We mix these cells in a spheroid. This is a tumor spheroid, and identify individual cells in the spheroid that have different reporters as well. We can spatially separate them and spectrally separate them. And because we're not, let's say, using lasers, we can image over many, many hours because it's just a chemical reaction. So that's the beauty of this, is that this luminescence property lasts for a while, so you can see them over minutes or over many hours. If you add the substrate, and they'll emit again. So here I'm showing you two color combination in the same cell. See if this one runs. And we have the intensity images here on the top. And we can spectrally separate out these two schemas. One is the GNL, which is the cytosolic reporter, and this is the H2B, which is a nuclear reporter, YNL. And then we can image that over time. So these are collected over time, and you could see the spectral separation of those reporters in the same cell. And of course, the idea would be to use reporters that actually measure metabolites. So if they bind to the metabolites of interest, we can spectrally separate those, calculate the threat efficiency, right? This is to undergo some sort of conformational change. And then depending on the distance between the donor and acceptor, that efficiency will tell us how many metabolites there are in the patient, for example. So we could do this. This is a donor and acceptor Brett pair, and calculate the Brett efficiency over time. So in this case, this is cell number and then the Brett efficiencies. So some cells undergoing more Brett, less Brett, and so forth, depending on the state of the cell. Okay, so what are we doing currently is now we're engineering a better phaser bioluminescence 2.0, or I guess this is 3.0 is their third construction, where instead of using two cameras, we're losing a lot of light. We decided to use just one camera and collect three reference windows, three windows. One is the reference signal, that's the white part here, and is the sine filter, and now there's the cosine filter. And then we can transform that into the phasers. And now if you're interested in the formula, we're calculating the cosine for the G coordinate, the sine for the S coordinate, and we get the phaser position for each one of those. And so this engineering system here is going to be, we're writing the paper now, and you have all the parts list. So if you want to build your own detector and spectral bioluminescence, you can build that. And we've also shipped it. We shipped out the part list and the instructions to a offsite lab that has built it themselves. And they're able to see the bioluminescence reporters. So we know that the protocol is good. It works. People understand it. Part list is easy to buy parts from. And the imaging pipeline for this particular setup is that even if you have very weak, or in this case, you have very bright signal, you can use cell segmentation using cell pose or whatever one that you want to use. There's different variants of identifying objects, so you can number each cell. And then we go from something like this that is very, very distributed on the phasers to having a clustering analysis. So you can, I don't know if you did this already in the in the program, but you could say, what's the average phaser position for that cell, and you get one spot. What's the average position of that cell, and you get one spot. right? So that way you can start to segment out individual cells and identify unique clusters. Okay, so the one camera system, we swapped out a Hamamatsu camera, which is a very sensitive camera, for a Nubu camera, which is much more sensitive, believe it or not, even though, you know, Hamamatsu claims to be the best. There's another camera that's actually better. For our purposes, it's called the Nubu camera, and we've identified, this is very weak. This is the luciferin I started with explaining to you. Luciferin is very, very weak. You can see that's very hard to tell the background from the cells, but we've been able to detect it. This was not even detectable with our turf, let's say, first setup, because we're splitting, and we can see enough signal. All right, so I'm going to shift gears to laser-based imaging. What we're doing currently in my group in my lab is inspired by mitochondria. So I'm interested in mitochondria because they have a lot of information that's very valuable. You probably see mitochondria images in the autofluorescence signal from FLIM. You can also label the mitochondria, and this is a project that was done by one of my grad students, Austin, whose task was to actually track mitochondria. And believe it or not, he was having a very hard time. And the reason for that is because mitochondria, you can label them, you can identify them, but they're stringy and they overlap. And you have some background in your cells. And so he was having a hard time actually tracing the mitochondria. So he decided to write a program. He called it Mitometer. He used diffuse background subtraction, which he borrowed from astrophysics, looking at galaxies and the stars, and subtracted the background, the diffuse background. That's what it looks like after the subtraction. He did a Gaussian filtering. He thresholded the mitochondria, and he got the final images that isolated the mitochondria. And using this, he was able to segment each individual mitochondria and follow them over time. And he was able to actually see if the mitochondria is undergoing fission, fusion, and so forth. We can also do this in organoids. So these are three spheroids. These are glioblastoma cells. You can see the mitochondria there. This is before Gaussian subtraction or filtering. But just suffice to say that once you do that, you are able to look at the differences in parameters of mitochondria. So this is, for example, the distance travel between mitochondria that are in cells that are grown in stiff hyaluronic acid or soft hyaluronic acid. It actually makes a difference of mitochondria dynamic. So in AH, which is stiff hyaluronic acid, the distance travel is very short. And soft, maybe at least, the distance travel is very long. There's a very striking difference there. The directionality also matters in terms of surfaces. And of course, you can measure the branching of the mitochondria. So the higher the solidity score, that means the more it looks like no branching, the less it looks like one single object. So the lower the score, meaning more branches. So they do have striking differences as well. And then the length of the axis is very even.
Mentioned this is a spheroid, a 3D object. Are you tracking it in 3D?
Mm-hmm.
That's great.
Yeah, we track those in 3D. And go to GitHub. We have Mitometer available on GitHub. I'm just checking to see if you're awake. I'm so glad you're laughing. On GitHub, and actually, Austin works at Calico. They have very nice funding if you want to work in a spectacular place. I highly encourage you to apply to Calico. And he has now a new computation that's much more superior than this. It's called Nelly. So if you're interested in this code, or I don't know if he's made a very fast plugin for images, but it's called Nelly. Okay. So what have we been continuing with this inspiration of mitochondria is looking at every organelle in the body, every organelle in the cell. It's like looking at every organ in your body, right? So if you can consider the nucleus, it's like your brain. The endosomes are like your kidneys and your stomach, because they do a lot of nutrients uptake, and kidneys also filter things. Mitochondria is where energy is stored. That's like the heart of your body, the heart of the cell. And then of course, like lungs, liver is like lipid droplets. This is the storage for fat. And then the bones are your microtubules inside the cell. So what if we can look at every single organelle simultaneously in the cell all at once? Can we see all of these beautiful parameters change as a function of perturbation? So how do we do this? Of course, for me, the mitochondria is the heart, so it'd be the center. We've been able to actually do this using 7 color dyes. It's a cocktail of different dyes that we've isolated the exact concentration and the minimal amount to stain the cells, live cells. They're alive, they're not dead. So you can do easy 3-color imaging, but when you get to 7 colors, that's a challenge, especially when you have huge overlaps. We're using a commercial microscope right now. It's a 32-channel array detector. It's not even the best detector, but that's what we got. So we're using this LSM 880 microscope with 32-channel array. And here are our function dyes. So we have lipid blue for lipid droplets. We have nuclear stain hooks. We have Bodipi for Golgi. We have rhodo green for lysosomes, mitochondria. TMRM is a membrane potential dye. We have lysotracker red, tubulin. And so each one of these dyes tells us the functional readout of those organelles. How do we do this? Well, it's hard because some of these dyes overlap, grossly overlap. So it becomes very challenging. So we need a new way to analyze the data. So I already showed you the 3-color, let's say fractional contribution. What happens when you have 4 components? I don't know if Alex talked about this, but he actually has his paper here referenced because he was the one that did 4-component separation. You can do 4 unknowns. You get 3 equations. They're underdetermined system. It's infinite solution. So just by using the fractional combination of those 4, it will be very hard to solve how unmixed up. So we need to go to higher order harmonics. The second harmonic solve 5 colors separation, and then we go up to the third harmonic to solve for 7 components. So in these equations, we have 4 unknowns, we have 5 equations, we have an overdetermined system, we have the exact solution. So we're able to separate out 7 colors. And I think that's as far as we can get. So unless there's something else I'm missing, but we have these targets. This is just one example. So for example, our target is DNA, that's the nucleus. What are we going to see? We can see the nuclear shape, the morphology of this structure, and then the function, chromatin compaction, right? So we have all of these parameters. We have a data-rich image that we can identify. Mitochondria, we have network organization, we have membrane potential, lysosomes, microtubules, and so on. Right? I'm not going to list them all, but the idea and the concept is a omic approach to organelle function for each independent cell. And so we have the image. We can use machine learning to become more efficient. We can segment each individual cell. We have the image of each cell here with the different colors. This is just 4 for an example. Lysosomes, mitochondria, lipid droplets, and membranes. And then we can do a single cell list of features. And we just got this paper accepted in Nature Methods, and we're able to identify hundreds of features from these kinds of data. So what kind of features? For example, you have here the whole cell that gives you the features of the TMRM, which is mitochondria. So you get the mean, the median, the sum, the max entropy, the variance. Just for TMRM label, you have organelles. These are another feature of TMRM, you get the morphology and you get the colocalization. If there's anything that colocalized, like maybe the mitochondria also interacted with the lysosome. We can actually calculate the distance between organelles because we can do image correlation spectroscopy and determine the distance of this.
One of the things, at least with the lifetime work then they went through the lab with Alex that they saw was like you need kind of like a calibration measurement off of some known, I forgot what starts with a C, I think.
Oh,
And-
like coumarin.
I don't remember what's coming, but basically then, so the lifetime of that, so then it can calibrate the image. In this case, is it simple enough just to have the spectrum of the dye, or you have to have the dye like
It
that'd be a
requires,
cell label dye so that
Yeah.
any shifts that happen, you have to take that into account, or?
no. So it depends on the state of the dye. If the dye shifts in spectra,
Yeah,
then that's kind of a problem because it could superimpose with some other color that you have in your, none of these shift in spectra. So that's the first thing. They're functional due to intensity levels. So we have, we have to incubate so that we make sure we have a completely, let's say, stable system, right? Steady state. You don't want something to be more bright or less bright because you like just labeled it, right? So everything is steady. So the functional, you have to go through like each one of the dyes and it tells you the function. But yeah, we don't have to worry about those things like referencing with coumarin because that tells us the instrument response function. In this case, it's just the spectra. So you have hundreds of these parameters. This is an example that we did this short time collaboration with Anne K. Love's lab, and she's working on a wild type cell and a mutant cells. And so we are looking at hundreds of these features in the differences between these cells. And when we found that, for example, there's the feature selection here for lipid storage was very good differences between the two cell types, the wild type and the mutant, but not so different in the lysosomal pH, for example. So we would rank this in high ranking for differences in feature, and this would be lower ranking because it looked very similar. So then you can map these in a UMAP and then see the difference between wild type and this mutant cells. And then if you look at the differences, this is over 4,000 cells that were imaged. You can get the agglomeratic clustering with the different phenotypes for these particular cluster fractions. So for example, cluster number 3 is more present in wild type, while cluster number 1 is more present in the mutant, right? And you can go back and identify what those were specifically in clustering. And was it more lysosomal? We have the fraction of each one and how they contributed to the differences for each one. So this would be an example of how the pipeline would be done. So with the 7 colors, so this is the first project is called Espresso. That's the technique. But with the 7 colors, which is what we're finishing and doing the time lapse, is what we're interested in seeing the effect of chemo resistance. So you really want to identify the features that gives us the phenotype of chemo resistance, and we can make predictions based on which ones are going to be more chemo resistant, less chemo resistant, and cancer cells, specifically breast cells. So we've changed in this conditions, these just changed different conditions of the cell. So this is not chemo resistancy. This is just doing a control change of perturbation in the cell, and they cluster into each one of these different perturbations that were done based on spectral features and phenotypes. Okay, so that's just kind of, this is unpublished, just to give you a glimpse as to what we're working on now. And we're working with machine learning, computational neuronal network scientists who are working with us to get these features identified. Okay, I don't know how much time I have. So I'm coming back to this slide. You tell me. Okay, so I can give you some highlights on some other work that we're working in our lab that has to do with installation, but let me just thank all the people that worked on those projects that you just saw. My former postdoc, Lorenzo Scipioni, and my current student, Song Ning, her thesis project on the seven color. And then Julia, who did a lot of this imaging. And Austin's here, he did the mitochondria. Oh, and I just wanted to give you a brief, if you want to copy this and share it to people you know. Our advanced imaging workshop in January 12th to 15th here at Irvine will be in that side too, or LICT. So we'll have that with, together with the first week. Okay, so as I just, I want to start off with this quote from Erwin Schrödinger that said, "How can the events in space and time which take place within the spatial boundary of a living system be accounted for by physicists and chemistry?" So essentially, he was talking about how can we measure, right, protein dynamics in space and time. So there's a lot of techniques that have been out there in the literature to do this, starting with single point fluorescence correlation spectroscopy that was developed by Elliot Elson and Watt Webb. And I guess that was a Watt Webb student, Elliot Elson, who's now retired. So it was many, many years ago. And then Enrico's group in Urbana-Champaign developed the scanning FCS system to account for many points, not just one single point. So it's called scanning FCS because you can take the laser beam and drive it in an orbit. Then Niels Peterson and Paul Weisman actually came out with something very innovative called image correlation spectroscopy that enabled them to calculate the spatial position, how big objects are in a given space. But they didn't take into account the time. So later on, Claire Brown, Niels Peterson's group, came out with temporal image correlation spectroscopy. So that's measuring the time correlations along many, many frames. But at that time, those frames were slow. So you could only measure things that were moving from frame to frame to frame. And then Paul Weisman went on to open his own lab, and he started with the development of spatial temporal image correlation spectroscopy that enabled you to look at changes of velocity inside the cell. And his grad student, Elvis, who's now in Australia, came out with velocity landscape correlation, and then they came out with K-space, which is the inverse space correlation spectroscopy, which is really cool. It's just very difficult to use. Nobody uses it too much, but it enables you to kind of isolate the frequencies of the diffusions. So what do we need all this for? Well, for our purposes, we're interested in phase separation. This is liquid-liquid phase separated states. And you can think of a system where you have molecules that then are phase separated, they undergo some coalescence. This is membraneless, right? They call them membraneless organelles. So these are structures that form without any particular, let's say, holding of anything else. And particularly, this happens in a very, very sharp transition, right? So you have this phase transition that happens in a very, very fast response. And it could also happen with a slow response, but those are minutes to hours, right? And this uses a lot of energy, whereas this does not. This is kind of no energy is used. So you have the phase separation states, you have coalescence, where these phase separation states come together and form one unit. There's a lot of different hypotheses that there's some sort of transportation of molecules going into the liquid-liquid phase separated states. There's also the sharing of molecules across two of these isolated phase separation states. And then there's something called liquefaction, which I always have a hard time remembering. I guess when solids turn to liquid is what happens. So let's see what I have here. most of the cases when this occurs is because the proteins that are forming these liquid-liquid phase separated states, they have an intrinsically disordered domain. Right, so you have these molecules that have these domains that don't really fold into any particular standard shape, alpha helix or beta helix or whatever. They're just kind of floppy kind of structures. And so they have a higher tendency to form these phase separated states. So there are many ways to measure phase separations. You can measure the FRAP. That means that fluorescence recovery after photobleaching. You bleach that spot. You wait for molecules to reoccupy that region, and you can measure the time that it takes for that to happen. You can use fluorescence correlation spectroscopy. You can use microbiology, laser tweezers. Anyone in the Beckman Laser Institute done the tweezers, phase separated states? Measure like the viscosity of those structures. You can also use fret sensors or dextran or nanoparticles. Okay, so what is FCS? Does anyone know? Okay, I'm just going to briefly tell you. Let's say that you are illuminating this whole room with the laser beam, okay? And that everything in this room is the excitation volume. So that's that blue circle there. As molecules transition in and out of that surface, they emit a photon burst. Now, they can emit a photon burst anytime along that beam, right, along that path. And eventually, you're going to build these photon bursts over time because your detector detects each burst. So it's almost like detecting single molecules. You're not visualizing them, they're not in, but you're detecting single molecular bursts. So now you have this data that looks like very spotty. What are you going to do? This data can be now, you can count the number of molecules. You could say how many molecules entered the spot. And this is a very famous experiment done by a scientist. His name was, let's say, call him Dr. Schwetzberg. His trusty worthy assistant was sitting next to him and looked into his new microscope that he could see gold colloids. In his observation volume, every time he saw a molecule, he would tell his trusty assistant how many of them he saw. So he had a metronome, a clock. So he knew exactly the time. So every time that metronome struck, he would say 1, 1, 1, 0, 0, 0, 2, 2, 3, 3, 3, 1, right? Whatever number came. Eventually his eyes got tired and he had to blink. So he would draw a little dash. But if you plot that number of, let's say, molecules there, you will understand that they have a Poissonian behavior. That means that, that they behave in that way. And you can calculate the average number of molecules that transition, similar to your photon burst. Now, another thing that you can get from this information is the time, how long it took for that molecule to diffuse across that volume. We know exactly what the volume is. We can calculate it, characterize it. And if we build the autocorrelation function, which is this, right here, this tells us that is a function of intensity times time multiplied by the intensity at a given time plus tau. So a little bit shifted in time divided by the average intensity squared, and that's normalized to 1. You build an autocorrelation function at very fast time scales. You're going to see the same molecule. Eventually, you're no longer going to see that molecule will decay away. So you can fit this into, let's say, a one species fit model, and the half of this curve will give you the diffusion time, the average time of residence. The molecule stayed in that position. You need many of these bursts. So this is called an ensemble averaging of molecular dynamics. So when people say, super resolution, people always say ensemble averaging. It's not very good, but ensemble averaging actually gives you a lot of information. The amplitude of this correlation function, which is called the G0, so when tau is equal to 0, is inversely proportional to the number of molecules you had in your observation volume. So you can actually calculate the concentration of the proteins there. So you have two equations or two extraction information, the autocorrelation curve, which is your number of molecules, the diffusion time. And what we care about is looking at this protein. So with collaborators from across the United States and the East Coast, we partnered to look at a protein they cared about called ACAP95. They have a protein that undergoes this let's say, liquid phase separation states. And this happens when the cell is going to divide. So it's very important for cell division. Don't ask me about the biology as much as I know. So they just know that something happens to the protein where it gets mutated in disease. All right, so what they did is they mutated this protein. And one of the ones that we looked at was the mutation of phenylalanine. So it went from its normal state, which is tyrosine, to phenylalanine. The control is YY, but the mutant is YF. And Y and F are tyrosine and phenylalanine. That's what those letters are for. So first we decided to do FCS and say, okay, can we tell the difference between diffusion of YY and YF? And so first, it does look very different, right? The curves look different. But the problem is that these structures move. They wiggle and they jiggle. So putting the laser beam in one spot, we found that the cell was like, or these things were moving. And then we had some issues with the decay. This didn't decay very long. So there's some issues with FCS. So FCS was not good. We decided to do something called RIX. And I didn't have time to explain this, but using one of those techniques I told you earlier, we developed the raster image correlation technique, and we did line scanning. So we took the laser beam and we said, okay, we're going to draw a line here as a function of time. And we're going to scan really, really quickly so that even if the cell moves, it's moving so slowly that we don't see that motion in our raster scan line. That's the fastest we can image. So now we're going to correlate in time and space this image. So we're going to perform a spatial temporal correlation of the intensity. If you want to know how this is done, I'm happy to tell you. But suffice to say that that worked because we're able to see the autocorrelation function of YY and YF, and they were different, statistically significant. So we know that the autocorrelation function yielded different macro scale. They trended for macro scale motion, so that didn't play any role. And we're able to see that the diffusions were different. How different? For YY, the diffusion was above 0.01. That means that faster than the proteins that were YF. The number of molecules were slightly different, right? It's spread between 200 and 600 molecules, where these were less. So indeed, the diffusion coefficient on average is faster for YY. There's larger fraction mobile than for YF. And so this data correlates with YFRAP that they had done earlier. So that was great because they were able to confirm that their FRAP experiments also worked. Okay, so my postdoc, Francesco, at the time was interested to understand the polarization of YY and YF to detect the aggregation of that could cause homofret. And I don't have time to go into all of these, what homofret is, but the only way to look at homofret is to look at polarization. So we took circularly polarized light. When the emission signal came back to our detectors, we put a polarizer such that we're able to see the parallel or the perpendicular emission of light. And so that enabled us to calculate the rotational diffusion. Of course, if the rotational diffusion is slow, it's almost immobile during the excited state. And the only way is to depolarize significant GFP by homofret. So can we tell the difference between YY and YF? The short answer is yes. So if you look at the color scale here, we see the intensity, right, of the protein, and then the polarization of the light emission in the parallel particular, this is high, this is low. So definitely there's a polarization effect here, and we look the R value and you see significance in the change of polarization here, and then we have a partition. So meaning in and out of the, in that. So the polarization R is significantly different, and the YF undergoes higher homofret. So this is more stuck. That means that most of those proteins are bound together. They're more stuck. So even though they both form condensates, their condensates are different, which is much more informative information for this biologist that we're doing this experiment. Okay, so I'm going to stop here and just show you the last thing is what we're working right now, because all that was data is published. We're working on a protein called WINK, and when you add salt to the cell, it forms these condensates. And you could see adding salt to the cell actually makes the cell shrink. So there's a lot of cell movement. It becomes kind of difficult problem to have. So we've been using image correlation spectroscopy, or RICS, to determine the diffusion of the protein. First, we have to calibrate the point spread function by fixing a no diffusion. This is just GFP without any CL. We get an omega squared, or 1 over E squared, which is 420 nanometers. That's substitution volume. And then once we fix that, we're able to look at GFP in the presence of sodium, and there's no difference. The diffusion is about 20, and with the presence of salt, about 23. And then, of course, this is WINK without salt. We looked at various different regions of the cell and looked at the autocorrelation function of those. They're about 2.3. When we add salt, this is what happens. We get a gross motion of the cell. Yeah, and I was trying to capture this before it formed the condensates, but it was really, really hard to do that because it happens in such a fast time scale. And you can see the difference before salt, after salt, and that's the bright field image on the cell. It's very challenging. So we're able to calculate the diffusion coefficients of these proteins. There's nothing in the nucleus. There's no autocorrelation function detected. But then, of course, in this coalesced state, we get slow diffusion, and we get a lot of this data. And of course, this is not published. This is just looking at the diffusion coefficients. This is before salt, we get an average diffusion of 2.55. After salt, it gets slow, and then it speeds up. And then at some point at 60 minutes, we get just the fast diffusion. So we're trying to understand is that once these proteins form this condensate, those little spots, they kind of get like stuck there. And then whatever is free in the cytosol is very, very fast, which means that it must have been bound to something that was impeding its diffusion. So when we told this to our collaborators, they're very excited because they said, oh, I believe that it's binding to something else. And they were doing some of those. So we've also done image brightness analysis to see if these things form higher order aggregates, and they don't. So they're all mostly monomers. The only time we see higher brightnesses is at the edges where things are moving, and that's a problem. If things move, then they'll give rise to variance, but we don't see, it's mostly a monomer, doesn't form any dimers or higher order aggregates. And that's about it. So we have a lot of work to do in this area, but those are our collaborators at UT Southwestern. That's all I have.
So the work that you did with using a lot of the dyes to take a look at the different organelles, different
Yeah.
components of the cell. I always had this question for fluorescence spectroscopy is every time you add a dye, does it change the biology of the
Yeah,
cell?
that's a good question. We are doing live MTT assays that tells us if the cell is having any problems with the dyes. So if the MTT assay shows us a perturbation in, let's say, viability, we'll see those changes. We don't see any changes so far as we've looked in the concentration of the dyes and the number of days that we've imaged the cells. So far, so good. They all seem to behave similar to physiological conditions. So we don't think that we're perturbing the system. Now, that said, I haven't tested if the combination of dye and laser are affecting the cell's health. Maybe, but haven't looked at that. Yeah. Any other questions?
There are several people here that are doing spectral imaging, like, so they're doing, just kind of classic, like, multispectral imaging.
Yeah.
And then they're taking, or hyperspectral, and then they're taking those images and then they're using absorption coefficient, I guess basis functions to try to fit different absorbers and things like that to the images. Can the phaser approach be adapted to reflectance images? And
Could be.
have you or has anybody tried that?
No, they haven't. That's a good point. I have talked to other people that were doing some other type of imaging. I'm trying to remember if it was MRI or something, or something that gave you a signal that was also can be converted to sinusoid. So it's almost like if you have a very sharp spectra, could you convert that into phaser space? And you could. So you could think that these kind of spectra can be converted into phaser space. I think definitely yes. You just have to work out the math for that.
I guess a follow-up question, or this I will not follow up. This is a different topic. You talked about the separating out like 7 fluorophores, which is pretty cool. And so just wanted to clarify. So when you said higher order harmonics, so that's just going from like fitting 1 cycle of the sine wave to 3 cycles of the sine wave.
Yes.
And
Yeah.
okay, so it's as simple as that. And then I think you said 7 was like a limit, if I remember.
I think we could
And-
only go up to the 3rd harmonic
Yeah.
so far, but due to the math. So we have to solve for 5 equations at that point. We haven't, first, I'm not sure that we can add more dyes
Okay.
to the system. So we haven't tried going to 9, 10 dyes. Oh, and one thing I forgot to mention is we're only using 1 wavelength to excite all these dyes. We're 7 colors, 1 laser, and 1 spectral detector simultaneously. So you don't have to use multiple lasers. Yeah, but that's a 2-photon laser, right? So we're using 2-photon excitation to excite the 7 colors.
No, that's impressive. Does the when you go to the higher order harmonics, does the signal to noise start going
A little
worse?
bit, yeah.
Okay.
Yeah. You need very good signal. Otherwise, it's like you just get a very broad spectra and you don't know where it came from exactly. Yeah.
Okay.
Yeah.
Yeah, I've just been thinking in my head, just kind of thinking, going through all the projects I know that everybody here is working on. I'm just thinking, is there a way something to apply it to in individual projects, like thinking about like photoacoustics and then just like the time resolved
Mhm.
measurement you get. Could you apply it there to try to help do some form of segmentation? Or I mean, it's just speculation. I don't know, but it seems like there's a lot
Could.
of interesting things that could happen with
Yeah.
at least from a mathematical perspective, pretty simple math.
Yeah. Yeah, sure. Yeah. Any other questions? I think everyone's tired. Had a long day today. But I'm available via email if you want to email me or you want to chat later. I'm happy to do that.
All right. Thank you so much, Michelle.
You're
Really
welcome.
appreciate it.